Welcome to my presentation. Today I am going to present a paper named a hierarchical bidirectional GRU model with attention for EEG-based emotion classification. I am H.M. Sajidul Haq, Assistant Professor, Department of Computer Science and Engineering at University of Chittagong, Bangladesh. My presentation has 7 sections. Firstly, I will introduce the paper, then I will say something about the motivation of this paper, and then I will state the problem which state the problem inside the paper, and next I will explain the objectives of the paper. And in proposed model section, I will explain the crucial part of this paper, which is based on the model or a scheme of for human emotion detections. And in the experimental result section, I will show the experiment. I will show some results on some data set, and finally I will conclude my speech. We know the activity of human emotions is closely related to the activity of neuron of our brain, and the activities of neurons can be represented as electroencephalogram EEG sequence, which consists of physiological, psychological, and pathological information. These type of signals can be used to read human mind for some control purposes. By reading the human minds, by reading the EEG signals, we can detect human emotions easily and classified, and that can be used in very sophisticated applications for making important decisions. And this is the motivation of this paper. We know that EEG signals has some inherent properties. For example, EEG signals are highly time independent, non-static, and inclined to noise interference. And these properties makes to extract precise key time points from a sequence which is related to the emotions difficult. And this is the great challenge. And for this reason, this is the great challenge to build a model for forecasting human emotion accurately. And this is the main problem of this paper. The main goals of this paper is to encode EEG signals into the model, and secondly, to build a model based on hierarchical attention mechanism with GRU model for human emotion detections. And finally, the model has been tested on benchmark dataset for measuring the accuracy. Now I am going to explain the crucial part of this paper. In the proposed model of this paper consists of 2 levels of processing. Firstly, the EEG signals are processed in the epoch level, and secondly, under these epochs, the samples are processed. The samples of EEG signals are processed. Here, the epochs are named as E, EJ, and where the samples are represented as X, I, J, T. In sample level processing, there are 2 parts. First part is sample encoder, and under the sample encoder there are multiple operations. And the first operation is to extract the features from samples and which are entered into the GRU cells And there are two types of GRU cells. One is forward directed GRU cells, another is backward directed GRU cells. What is GRU cells? GRU stood for gated recurrent unit, and this model is very popular for analyzing the sequence data. The features are entered, the features of samples are entered in both cells by weighting two weight vectors W1 and W3. I'm mentioning one thing here. The dimension of the length of the features are C, length of the features of samples are C. And the internal structure of the GRU model is like this, and where the mathematical equations is also shown in this slide. Here, there are four parameters. One is HT, ZT, RT, and HT-tel, where HT is the new hidden state, HT-tel is the candidate hidden state, and ZT and RT are the updated gates and reset gate respectively. And every GRU cell produce one state, and this is mentioned here, HJF or HJB. And each cell is forwarding the state information to the next cell by weighting some values, and in the forward direction the weight value is W2, and the reverse direction, in the backward direction, the weight value is W5. Here I'm mentioning one thing. The forward directed GRU cells are used for processing the past informations of the EEG sequence, and the backward directed GRU cells are used to process the future information of EEG sequence. So after computing the state of forward and backward directed GRU cells, we have the model will combine those values into another annotation vectors, which is called HJ1. And so, during the concatenation, the values comes from forward GRU cell and backward GRU cells are weighted with two variables, which is W4 and W6. And this is the last operation of sample encoder sections. And the output of these sections will go to the next level of operations, which is called sample attention. And under this sample attention, there are three operations will be performed. And the first operation will be performed through some multi-layer perceptrons, and where the activation functions are 10 A's. And the annotation sample annotation vectors are weighted with WS to the perceptron and produce the value UJ1, which is combined with the sample level context vector into the softmax to compute the importance of each sample. And the softmax produce the probability which is multiplied with the sample annotation vector Aj, and by combining those values, we have got a epoch vector which is passed to the epoch level processing. Here the epoch vector is Ej. After finishing the epoch level, after finishing the sample level processing, and then the epoch level processing will be started. I have already mentioned the epoch level processing also divided into two parts. First one is sample encoder. Second level is first level is epoch encoder, and second level is first step is epoch encoder, and second step is epoch attention. The operations under the epoch encoder is completely same as the sample encoder. Here it has also a GRU, forward directed GRU cells and backward directed GRU cells. The difference is that the weighted vectors are completely different. Where in epoch encoder, the weighted vectors are named from W7 to W12. So after finishing the epoch encoder, we will get another annotation vector for epoch, which is mentioned as H1 up to HP. Since it has P epochs, that's why it has the length of the epoch vector is P. After finishing the epoch encoder, the output of the epoch encoder will go to the epoch attention. And I have already mentioned the set of operation inside the epoch attention is also same as the attention sample attention. But here the one important thing is that here the importance of epoch has been calculated by combining epoch level vector. So inside the softmax operation, here the output of the softmax operation is also a probability, and which is also combined with the epoch annotation vector H1 up to HP. And this is the equation which will be used in the epoch attention. If we see very carefully, then we can see that the equation of the epoch attention and the equation of the simple attention is very similar. So after combining the probability comes from softmax with epoch annotation epoch vector, we get a new vector which is called sequence vector and named V. And this sequence vector will be used for classifying the human emotion. And the equation of the classification of the human emotion is like this, where WC is the weight vectors of sequence vectors, and VC is the bias values. And this is the all over the proposed model for of this paper. And we know inside this model we have to update our weights. So to update the weights, we need a loss function, and the loss function is shown in this here as E. So this is all about the proposed model. And then I am going to explain some experimental results. The proposed model has been applied on the benchmark data set, which is called DAPE and developed by researchers at Queen Mary University of London. And after applying those data set, it has been seen that for 0.5-second segmented ECG sequence, the proposed model is showing the better accuracy, which is 69.3% than others models like GRU, hierarchical but non-attention HFB GRU, model, hierarchical but non-attention H-max GRU model, and best baseline LSTM model. And secondly, for 1-second segment EEG sequences, the proposed model is also performing better. And the accuracy rate is 67.9% for balance emotion and 66.5% for arousal emotion. Where we can see the rest of the model like LST model and shallow baseline model are showing lower accuracy rate. Now I'm going to conclude my speech. This paper has been proposed. This paper has proposed a very good model named hierarchical bidirectional GRU model with attention mechanism for EEG sequence learning for human emotion prediction. And this model, the whole model can be divided into two parts, two levels, one for samples level processing, another for epochs level processing. In the samples level processing, samples are encoded. On the other hand, epochs level processing, the epochs are encoded inside the processing model. And in both level, the attention mechanism is used. The importance of epochs and samples are aggregated by using the samples and epoch context where we have already seen. So this and we have seen that the proposed model outperforms the others models. And the future work of this paper is we can easily develop a robust applications which can interact, which can make interface between brain and computer. And thank you. This is the end of my presentations. Thank you. Thank you.